All right. So starting with the topic of mitosis and basically your meiosis. Okay. So when you talk about your mitosis and meiosis, you first need to remember that there are two types of reproduction that we deal with. We have asexual reproduction and we have sexual reproduction. Asexual reproduction is done when you have one single parent. There is one single parent, there is no gametes, there is no sex cells involved, no sperm, no ova, nothing. It's going to be from one single parent. It's going to be because of mitosis. That's very important. And the offspring is going to be genetically identical, which means whatever is going to be the genes of the parent, the exact same genes are going to be present in your offspring as well, right? When you talk about sexual reproduction, the sexual reproduction involves two parents. You need to have two particular parents. And when you're going to talk about it, you need there it involves the fusion of your sex cells or your gametes. How are the sex cells made? The sex cells are made by the process of meiosis. Okay, sex cells are made by the process of meiosis, and what is actually going to end up happening, that they're going to fuse with each other, and that is going to be known as sexual reproduction. In sexual reproduction, the offspring is going to exhibit something known as genetic variation. What is genetic variation? Genetic variation is when the genes are different compared to the parents, right? So because the genes are going to be different compared to the parents, and that's what you observe in animals, in humans, right? That the children are different than their parents. They have some characteristics that are similar, some that are different, and that is because of genetic variation. So to understand this, we have to go ahead and we have to talk about mitosis and meiosis. But before we can understand mitosis and meiosis, we have to understand what happens to the DNA inside our cells. So generally the DNA inside our cells is present in the form of a thread-like structure that is known as chromatin. Okay. Chromatin, when the cell is in, when you talk about the cell, when the cell is in a growth phase, when the cell is in a growth phase, the DNA is present in the form of chromatin. But when the cell is about to replicate, when the cell is about to divide, this chromatin is going to wrap around each other. The chromatin is going to condense to give you your chromosome. Okay. Now, how does this entire process actually happen? Let's start with our DNA. Our DNA is going to turn into chromatin. How? Our DNA is going to start wrapping itself around these proteins. Around these proteins. These proteins are known as histones. These proteins are known as histones. So what's going to happen, that the DNA starts to wrap itself, it starts to intertwine with these proteins that are known as histones. Now, what happens, that this entire structure, DNA plus histones, this entire structure, we call it a nucleosome. So a nucleosome is when you have a DNA wrapped around a histone. We call it a nucleosome. So the DNA is going to start to wrap itself around these proteins known as histones. Once the DNA has wrapped itself around the proteins, what's going to happen? These proteins are going to start to twist around each other. They're going to start to wrap around each other. They're going to start to, condense together, and they're going to come together to give you a very thin thread-like structure that will be known as chromatin. So generally, when you talk about it in your normal growth phase of the cell cycle, if you ever try to visualize your DNA, you will see it in the form of chromatin. But if you decide to see the DNA before the cell is about to divide, you will observe that the chromatin is going to end up condensing. It's going to end up wrapping. It's going to come together. It's going to all condense, and it's going to give you a chromosome. So a chromosome is going to be made when the cell is about to undergo cell division. When you talk about chromosome, now what are the different parts of the chromosome? First thing we have to realize is this. The chromosome is made up of chromatid. This entire this one thing, this is going to be known as a chromatid. Okay. And this, we have got two chromatids. Both the chromatids are completely identical to each other. Why do we have two chromatids? Because our cell is dividing. Since my cell is dividing, so it has to replicate. It has to replicate. So the chromatid will, for example, have the original DNA. And the other chromatid will have the copy of it. So that is what happens when the DNA replicates, when the DNA divides, you basically have two chromatids that are identical to each other. One has the original DNA, the other one has the exact copy of it, right? So when the cell is going to divide, one chromatid can go to one cell and the other chromatid can go to the other cell. So we call these chromatids, we call them sister chromatids because of course they are completely identical to each other. So we have our chromatids and the chromatids are attached to each other at the middle by something known as kinetochore. So we have a kinetochore and the kinetochore is basically, the kinetochore is basically going to attach the two sister chromatids to each other. It's going to attach the two sister chromatids to each other. Along with this, you have something else in the center. Okay? We have something else in the center. The thing we have in the center, if you look over here, this round thing, this is basically your centromere. Okay? So when you are going to go ahead and talk about it, one second. Okay, I'm sorry. One thing I want to clear up. The kinetochore is not the one that is attaching the sister chromatids. Okay, the centromere is the one that is attaching the sister chromatids to each other to give you your chromosome. What is kinetochore? Kinetochore is going to be it's present over here, right in the same area. The area is the same, but the kinetochore will allow the chromosomes to attach to your spindles. We will study about the spindles when we do mitosis and meiosis. But we have these spindles, we have these microtubules that help in the separation of chromosomes, and help it separate into two. So what do we have? We basically have the kinetochore. So basically kinetochore will allow the chromosome to attach to what? To attach to your microtubule, to attach to your spindle, while the centromere will hold the two sister chromatids with each other. Along with that, there are two arms. One is known as the short arm and one is known as the long arm of chromosome. Any confusion so far? Anything you want to ask? Okay. Then what you can see over here is basically known as a karyotype. What is a karyotype? Karyotype is basically we can stain the chromosomes and we can visualize them. So generally speaking, you know that in humans, we have 46 chromosomes. 46 is the total number of chromosomes. If I decide to count them in the form of pairs, so there are 23 pairs, or if I count them one by one, then I have 46 chromosomes. From those 23 pairs, 22 pairs are considered to be autosomes and one pair is considered to be the sex chromosome. So the X and Y chromosome is going to be the sex chromosomes, while the rest of the 22 pairs are going to be your autosomes. And when we are going to go ahead and we're going to talk about it, Karyotyping is very helpful because it can let us observe the chromosome and let us diagnose any, chromosomal disease that the person can have. Now let's talk about our cell cycle. In our cell cycle, there are two major stages. We have interphase and we have mitosis. Okay, we have interphase and we have mitosis. What is interphase? Interphase is basically the space in which the cell is not dividing. There are three stages or three steps in interphase. G1, which is known as gap 1. We have the S phase, which is also known as the replication phase. And then we have the G2 phase, which is also known as gap 2 phase. What happens in the entire process? Let's discuss. In the first phase, that's the G1 phase or the gap 1 phase, what happens? That basically growth occurs. The cell is just growing. Let's say you have a pancreatic cell. So it's going to be making insulin, it's going to be doing its normal function, it's going to be making ATP, it's going to be doing whatever job it does, it's just going to be growing normally. Now, so it's completely normal, just the cell is growing, cell is making more protein, cell is making more lipids, cell is doing its function, it's making energy, it's making food for itself, whatever. Then you go on to the S phase, which is the replication phase. This is the phase in which the DNA actually replicates. This is the phase in which you end up getting a copy of the DNA. And after the fast S phase, you have the gap 2 phase. What happens in the gap 2 phase? The cell prepares itself for cell division. So any other stuff that is needed, for example, we need centrioles. I'll tell you what centrioles are. We need centrioles for process of cell division. So the centrioles will replicate, they will divide. Your other organelles. The other organelles will also start dividing and they will start getting ready for the process of cell division. After that, you will end up getting your mitosis. And I will discuss with you what actually happens in mitosis. Before we talk about what happens in mitosis, we have to talk about our checkpoints. Between each phase, we have certain checkpoints. What is the function of these checkpoints? To analyze the DNA and to make sure that our DNA has not mutated. So for example, once the cell is about to complete gap 1 and it's about to replicate, it's about to go towards S phase, what will happen? There are these checkpoints. The checkpoint is going to analyze the DNA and it's going to make sure that the DNA has not been mutated. If the DNA has been mutated, the cell will instantly shift towards apoptosis. What is apoptosis? Programmed cell death. So the checkpoint is going to be like, listen, we've had a DNA mutation and there's no way we can go on forward with this. This is abnormal now. We need to terminate, destroy the cell. So the cell will instantly go towards cell death. Let's say that at G1, everything was A-okay. So it will say, okay, DNA is fine, everything's fine. Let's go on to the next step. That is DNA replication. After DNA replication is done, there's again going to be another check to make sure there has been no mutation whatsoever. And if there has been no mutation whatsoever, then it's going to say, listen, we are good to go to the next stage. Or it's going to say, no, we have been compromised. DNA has been mutated. Let's go towards apoptosis. This is how the body protects itself from development of mutations and development of cancer. And if this checkpoint fails, if this checkpoint fails, that can lead to cancer. Because if there is a DNA mutation and the checkpoint has failed, the mutation will pass through, the cell will divide with the mutation, and that can lead to abnormal growth and cancer. So this is one of the reasons of how cancer actually happens. So now you can go ahead and ask me and you can say, okay, miss, but how does this checkpoint actually work? This checkpoint depends upon two proteins, a protein and an enzyme. The proteins are known as cyclins And the enzyme is known as cyclin-dependent kinases. So what's going to happen is if everything is A-OK, if everything is going to be A-OK, what's going to end up happening? The cyclin will bind to the enzyme. The cyclin will bind to the cyclin-dependent kinases. And when the cyclin is going to bind with the enzyme, when the cyclin is going to bind with the enzyme, it is going to start producing changes. For example, it will work on the nucleus and it will tell the nucleus, we are good to go ahead and start making the chromosomes. Or it will say, we are good to go, you can start breaking down the nuclear envelope for cell division. So this is what happens in your cell cycle. Now, let's take a look at our mitosis. In our mitosis, what happens? There are four major phases that you need to know about: prophase, metaphase, anaphase, and telophase. What happens in prophase? In prophase, you start by breaking down the nuclear membrane, the nuclear envelope. So at this stage, let me tell you, the chromosomes are ready. The DNA has replicated itself. The DNA has divided itself. Your chromosomes are ready to go. Okay, so if you're going to look at your nucleus, what are you going to observe? Lots of darkly staining chromosomes. The second thing you will observe is that your nuclear membrane is going to be breaking down because you need to now start the process of cell division. The nucleus will just stop you. So you need to break down the nuclear envelope. And your centrioles, these are your centrioles over here. The centrioles are needed for what? They are needed for making something known as spindle. That is what centrioles are. So you have centrioles, and these centrioles are going to start to move to the opposite side of the cell. So centrioles go to the opposite side of the cell, the nuclear membrane disappears, and the chromosomes appear condensed, which means you move on to metaphase. In metaphase, what ends up happening is this, that at this point, look over here. The centrioles are on opposite sides. And over here, between the centrioles, you have your spindle fibers. The spindle fibers are made up of a protein we call microtubule. So the spindle is present, the spindle fibers are present. What's going to happen? The chromosomes are going to arrange themselves right in the center of the cell in one straight line, like this. The chromosomes will arrange themselves right in the center of the cell in one straight line. Okay. This is by the way, the middle of the cell is known as your metaphase plate, or it's basically known as the equator of the cell. So right in the middle where you have your equator of the cell, where you have your metaphasal plate, that is where the chromosomes are going to arrange themselves. This is known as metaphase. In anaphase, what is going to end up happening is this, that your mitotic spindle is going to start to contract. The spindle fibers, the microtubules are going to start to contract. And as they're going to start to, contract, what is going to end up happening? What's going to end up happening is this, that your chromosome is going to break into two. Remember, I told you the chromosome is made up of sister chromatids, right? And they were completely identical to each other. So the chromosome breaks into two, it separates into two, and they are pulled on the opposite sides. They're going to be pulled onto the opposite sides of the cell. As they're going to be pulled to the opposite sides of the cell, what is going to end up happening? That is anaphase. So in anaphase, the chromosomes are going to break at the center, and the chromatids are going to be pulled to the opposite sides of the cell. This will be followed by telophase. In telophase, what's going to end up happening? That the nuclear membrane will form once again. If you take a look, the nuclear membrane is going to form once again, and the cytoplasm is going to start to divide. The nuclear membrane is going to start to form once again, and your chromosome is going to also start to again turn into chromatin. So chromosome has done its job. It's going to start turning into chromatin. nuclear membrane will form and your nucleus is now complete once again. The final stage is cytokinesis, in which the cytoplasm and everything else is going to divide and the cell will break away into two. This is the process of mitosis. Any confusion so far? Is this all making sense? Okay. Now what we have to cover is we have to talk a little bit about cytokinesis, how the cells are breaking away into two. Let me first tell you how cytokinesis happens in animal cells. In animal cells, the cytokinesis is actually pretty simple. Okay. What happens is that you end up getting this sort of a band. You end up getting this constriction right in the center, right? You end up getting this constriction right in the center, and the restriction will keep on getting narrower and narrower and narrower, and the cell breaks away into two. So pretty simple. In animal cells, it's really simple to understand. In plant cells, there's a little bit of logic to it. What's going to start to happen is this, that you have your, this is basically what is happening. This is your nucleus. It has formed once again in telophase. Now, what happens is that, can you see these lines? These lines are basically your remaining spindle fiber. So not all of the spindle fiber breaks away. Some of the spindle fiber gets left behind. And what happens that these spindle fibers guide the Golgi vesicles and ask them to come to the middle of the cell. So the spindle fibers will guide, they will tell the Golgi vesicle, listen, this is where I am in the center of the cell. So you need to come and you need to come right over here. So the Golgi vesicles will start to accumulate in the middle of the cell. Then the Golgi apparatus, as they start to accumulate in the middle of the cell, they will start to fuse with the cell surface membrane. They will start to fuse with each other firstly. They will start to, so you have small Golgi vesicles, they will start to fuse with each other to give you the large vesicles. And on top of that, it will also start to fuse with your cell membrane over here and over here. The Golgi vesicles are going to keep on enlarging and they're going to keep on fusing together to make a cell plate like this right in the middle. And what is going to end up happening that between these Golgi vesicles, what's going to happen? Cellulose is going to be deposited. Between these Golgi vesicles that are fusing with each other, cellulose is going to be deposited, and as a result, you are going to end up, the cell will end up dividing into two. In the regions where the Golgi vesicles do not fuse, in those certain regions where the Golgi vesicles do not fuse, we have gaps. Those gaps are known as plasmodesma. And plasmodesma are gaps that allow the cells to communicate with each other, that allows the cells to basically talk with each other. Okay, so this is how cytokinesis is actually happening. Then we have to talk about how we can observe mitosis. So we can observe mitosis simply, first of all, by preparing our slide. So how do we prepare our slide? We can basically go ahead. So we generally, when you do the experiment, by the way, if you want to go through it, Manur, the core practicals are over there on the website. Okay. The core practicals and unit 3 past papers are there on the website solved. So if you want to go through them, you can go through them, and your core practicals are over there as well. So over there, I have discussed in detail how we do it. But we basically take a growing tip from a root, and what we do it is that we go ahead, we slice it, or you can squash it. And then what you have to do is you have to stain it. So there are multiple stains we can use. For example, we can use orison, acetic acid, we can use methylene blue. All of these stains can be used to visualize the chromosome. So what you can go ahead and do is that you calculate the mitotic index. So mitotic index is calculated by counting the number of cells that are undergoing mitosis and dividing them by the total number of cells. So for example, this cell is undergoing mitosis 1, 2, 3, 4, 5, 6, 7, 8. So you count the number of cells that are undergoing mitosis, you count the total number of cells, and you simply divide the two of them to find out your mitotic index. So over here, for example, this is, what is this? This is anaphase, because you can see that the chromosomes are going to the opposite side. What is, again, this is anaphase. Again, this is going to be anaphase. This is also anaphase. This is metaphase. You can see that they have accumulated over here right in the center. Same, this is also anaphase. This is also anaphase. So what you're going to do is you're going to count the number of cells that are divided by the total number of cells, and you'll get your index. Now, let's go ahead and talk about meiosis. To understand meiosis, we have to first talk about your gametes. What are gametes? Gametes are, of course, your sex cells. When you're going to talk about your sex cells, in your sex cell, basically, you know two terminologies, diploid and haploid. Diploid is when you have complete set of chromosomes. Okay. And haploid is when you have only one chromosome from each pair. Okay. So for example, if I have one pair of chromosome number 1, so if I have both of them, that is going to be diploid. Or for example, if I talk about the sex chromosomes, so the sex chromosomes are basically, if I have a pair, if I have a diploid number, I will have both X and Y, or both X and X. In a haploid situation, I will only have one chromosome from each pair. So from the chromosome, from the sex chromosome pair, I will either have X or Y. Okay. So you know your normal body cells. By the way, the normal cells of the body are known as somatic cells. Okay. So we have somatic cells and we have gametes. So somatic cells are generally diploid and gametes are haploid because they have half the number of chromosomes. So when we're going to talk about it, in flowering plants, the female sex organ is the ovaries and the male sex organ is the anthers. The female inside the ovaries, you have ovules that are the female gametes, and on the anther, especially when you're going to talk about it, on the anther, we have our pollen that acts as the male gamete. In animals, we have testes and we have our ovaries. In the testes, you end up getting sperm, which is going to be the male gamete, and in females, we end up getting ova or egg. Now, how does meiosis actually happen? Meiosis takes place in two stages, meiosis 1 and meiosis 2. And when you're going to talk about it, remember this. In mitosis, we end up getting 2 cells. So one cell will give me 2 cells. But in meiosis, one cell gives me 4 cells. Okay. So let's go ahead and see what is going to end up happening. So when I'm going to start with meiosis number 1. The first stage of meiosis number 1 is known as prophase, right? It is basically known as prophase 2. Sorry, prophase 1. In meiosis 1, you will have prophase 1. Prophase 1, what is going to end up happening? Some of the steps are going to be the same. For example, breaking down of the nuclear envelope, that is going to be the same. The centrals will go to the opposite side. That is going to be the same. The chromosome is going to, the chromatin will condense to give you chromosomes. That is going to be the same. Now, one thing different that is going to happen in prophase 1 is known as homologous pairing. Those chromosomes that are homologous, they will pair with each other. Okay, they will pair with each other. That is known as homologous pairing. So for example, if you have two sets of chromosome number 1, right? If you have two sets of chromosome number 1, so both the chromosome number 1s will pair with each other. That just like that, if you have, for example, X chromosome and Y chromosome, so they're homologous, so they will pair with each other. Okay. Now, when homologous pairing is going to occur, something happens that is known as crossing over. What is crossing over? What happens in crossing over is this, that that when you're going to talk about it, what is going to end up happening, that these are your chromatids, right? So the chromatids of the two chromosomes will overlap with each other. They will attach to each other and they're going to overlap with each other. And as they're going to overlap with each other, they will break away and they will recombine. What do I mean by that? Let me tell you. This is my one chromosome, okay? This is another chromosome. So what is going to end up happening, that both of them are both of the chromatids. So homologous pairing is going to occur. After that, the chromatids are going to overlap with each other. After the chromatids are going to overlap with each other, the chromatids will break. What's going to end up happening? The reason that it generally ends up happening is that because this introduces genetic variation. Okay, if you've studied biodiversity and conservation, you know how important genetic variation is for the survival of a species. Because the more variation you have, the better your chances are of adapting to a new situation. So by this crossing over, what happens, that you basically change the chromosome. So instead of getting an identical chromosome, now the chromosome is a bit mixed up. So that brings variation. The genes are not the same. It changes up a bit. So that makes sure that the offspring is going to have genetic variation in it. So that offspring will have a better chance of survival. So that's why. So what's going to happen is that it's going to break away, and this is what you're going to end up observing. Like this. See, now a portion of the blue chromosome is attached to the red chromosome, and the blue chromosome has a little bit of red chromosome. So what is this doing? This is basically now this chromosome has changed. It's not the same anymore. It's not the exact same as the parents anymore. It's now a little bit different. So when the offspring is going to get it, they're going to end up getting a little bit of different chromosome. So that is going to make the offspring more variable. Their genes are going to be more different compared to the parents. So that's going to better increase their chances of surviving, because they are better able to adapt to different environmental changes. Making sense? Okay. Now crossing over is going to happen. So crossing over, homologous pairing, breaking down of nuclear membrane, centrioles will go to the opposite side. All of this will happen in prophase 1. Now we go on to metaphase 1. Metaphase 1, just like that, you have your spindles, and on the spindles, the chromosomes will attach themselves in the center. But this is where a different comes. This is my meiosis. In meiosis, the pairs are arranging in the spindles. Over here, they were arranging one by one. They were arranging in one single line. So one by one, the chromosomes were coming in the middle. Over here, the pairs are arranging themselves in the middle. Okay, the homologous pairing that took place, that entire pair will come and attach in the middle. So like in mitosis, you were getting one chromosome only. So over here, you will have two, because the entire pair will attach itself. So this is pair number 1, this is pair number 2, pair number 3, pair number 4. Just like that, the pairs are going to come and start to attach themselves to the middle of the cell. After that, everything is the same. After metaphase 1, you will get anaphase 1. So in anaphase 1, what's going to happen that over here you can see chromosome is not breaking. The chromosome is remaining completely joined up. It's remaining completely intact. What is happening is this, that one entire chromosome is going to one side and one entire chromosome is going to the other cell. Okay, so over there, what was happening? The chromosome was breaking into two, and the chromatid was going to one side and the chromatid was going to the other side. Over here, the pairs will simply separate because you were getting the entire pair over here, right? So the pairs will simply separate. One chromosome will go to the one side and the other chromosome is going to get to the other side. Okay. So what is going to end up happening after this? After this, we will end up getting telophase, and telophase is going to be exactly the same as your, you can say it's going to be the same as what? It's going to be the same as mitosis. Now we are done with the first stage of meiosis. Now after this, you are immediately going to move on to stage number 2. Okay, instantly going to move on to stage number 2. So by the way, let me clear one thing up. Let's say that our haploid cell, our diploid cell, the diploid cell had chromosomes. The number of chromosome was when you're going to go ahead and talk about it. So in a diploid cell, basically you are, for example, having the chromosome number of 2. So you can see over here, we're starting with 4 chromosomes. Why? Because the DNA has replicated itself. Because the DNA has replicated itself, that is why we're starting with 4 chromosomes. So at the end of meiosis 1, we have 2 cells and each cell has 2 chromosomes. Okay, 2 is the diploid number. Now, after this, immediately you're going to move on to meiosis 2. What is going to happen in meiosis 2? In meiosis 2, you're going to start with prophase 2. Prophase 2 is the same as mitosis. Exactly the same as mitosis. After that, you will get metaphase. Metaphase is also the same as mitosis. Meiosis 2 basically is mitosis. Meiosis 2 is literally the same thing as mitosis. Okay? What's going to happen? That your chromosomes in metaphase 2, the chromosomes are going to line up in the center one by one. Just like in mitosis, they will arrange in the center one by one. In anaphase 2, the chromosome will break in the middle and one chromatid will go to one cell and one chromatid is going to go to the other cell. And then in telophase 2, the cells are going to be made. So over here, remember at the end of meiosis 1, we got 2 cells. So over here, each cell is going to divide and give me 2 more cells. That's why the total number of cells I will get is 4. And if you look over here at the very end, each cell has only 1 chromosome in it, which is our haploid number because our diploid number was 2. So our haploid number is going to be half. It's going to be 1. So each cell now has 1 chromosome inside of it. So meiosis 2 is exactly the same as mitosis. Meiosis 1 is different. So meiosis 1 is basically what you have to learn. But for meiosis 2, whatever you have studied in mitosis, you can just copy that directly. Okay, making sense? Okay. By the way, another thing that we need to go ahead and talk about, another thing that takes place over here in your, so remember one of the things that introduces genetic variation is known as crossing over. There is one more thing that introduces genetic variation, and that is known as random assortment. What is random assortment? So basically, what is random assortment is that when your cell is dividing over here, okay, when your cell is dividing over here, the chromosomes are randomly divided. Okay, the chromosomes are going to be randomly divided between the two cells. That is known as random assortment. Okay, so you're going to end up with what? You're basically going to end up with random combinations or random distribution of your chromosomes. So that is known as random assortment. Random assortment is also important because it also reduces genetic variation. Because what combination of chromosome is, passing on, what combination of chromosome is actually passing on, that's going to be different for each cell. Okay, now we have to go ahead and go over our gamete formation. Have you gone over gamete formation in plants and in animals, this fertilization and all, and fertilization in human and fertilization in plants? Have you gone over it, Mahanur? Is this part, do you want me to go over this part? Okay. First of all, let's just do humans first, and then we'll do plants first altogether. I'm not doing gametes first and then fertilization. I'll explain to you just humans first, and then we'll do all of plants first. So in humans, we have we have our sperm and we have our egg. Okay. So in the sperm, we have three major parts. We have a head, we have a middle piece, and we have a tail. So we have a head, we have a middle piece in the middle, and we have our tail. When we talk about our head has one thing, of course, it has is the nucleus. The nucleus is very important because it contains the chromosomes inside of it, right? It contains the chromosomes inside of it that we need. It has the genetic information. Along with it, it also has something known as an acrosome. What is an acrosome? Acrosome is basically, it contains enzymes. It contains an enzyme, and the function of the enzyme is to destroy the protective layer around the egg cell. So the acrosome has enzymes in it that have the function of destroying what? Destroying the protective layer around the egg cell that is in the head. So we have the nucleus and the acrosome in the head. Now, at the very end, we have a tail. What is the function of the tail? The tail allows mobility. It allows the sperm to move up the uterus and go towards the fallopian tube. In the fallopian tube or in the oviduct, that is where fertilization occurs. Okay. So it allows the sperm to travel up the uterus to the fallopian tube for the process of fertilization. And right in the middle piece, we have an abundant great number of mitochondria. Why? To provide ATP so the tail can work. The mitochondria will make the ATP, so the tail can use that ATP and allow the sperm to swim up the uterus. For the egg, let's talk about the egg. So in the egg, at the very start, we have, in the very middle, we have our nucleus. And in that nucleus, we will, of course, have our chromosomes. We will have our genetic material. Outside the nucleus, we have our cytoplasm. And the cytoplasm is filled with yolk particles, like an egg yolk. So yolk particles. And the function of the yolk particles is to provide nutrition to the zygote. So in cases of fertilization, so if you remember, what happens is that once you have fertilization, the egg has to, you will basically get a zygote. And the zygote then has to travel down the oviduct and it has to implant itself in the lining of the uterus. Only after it has implanted itself in the lining of the uterus, you get the placenta and the umbilical cord. So up till that stage, up till it implants into the wall of the uterus, it needs nutrition from somewhere. So it comes from the yolk particles. From the yolk that is present in the cytoplasm, it will provide nutrition to the zygote. Now, of course, then we have our cell membrane, and outside the cell membrane, we have a protective coating. This protective coating is known as zona pellucida. It's known as zona pellucida, and it is present right outside the egg cell. And of course, these little cells you see on the sides, these are known as follicle cells. So the egg is actually present in the ovary, and in the ovary, it can be found in a follicle. So generally what happens is this, that when ovulation occurs, the follicle bursts and the egg is released. So the egg still has some follicle cells attached to it. So this is the structure of it. Now, what you are going to go ahead and talk about it, we have to talk about how does fertilization actually occur. Okay. Now, what's going to happen is this, that the sperm is going to enter into the uterine cavity, and it is basically going to swim up the uterine cavity up to your fallopian tube or up to your oviduct where the egg cell is present. Over there, the sperm is basically going to what? It is going to, it's basically now, before the sperm can do fertilization, it has to break down that zona pellucida. It has to break down that protective layer, because otherwise it will not be allowed to fuse. So the acrosome that is present inside the sperm, it will start producing enzymes. Those enzymes will start to break down the zona pellucida. And this is not something that is done by one particular sperm. Millions and millions and millions of sperm work together to produce the enzyme. The enzyme is known as acrosin, by the way. They produce the enzyme and they start to break through the zona pellucida. Okay, so this is known as the acrosome reaction. So the moment the sperm is going to touch the zona pellucida, the acrosome reaction is going to start. So enzymes are going to start to pour out of the acrosome, and they're going to start to break down the zona pellucida. Okay, now eventually what's going to happen, one of the sperm is going to manage to make its way through. One of the sperm is going to be able to make its way through, and it's going to penetrate the cell membrane. It's going to break through the cell membrane of the egg cell, and what is going to happen, it will release its nucleus into the cytoplasm. Now, the moment this happens, and the moment this is going to happen, you will end up getting another reaction that is known as cortical reaction. What is cortical reaction? Cortical reaction is basically going to happen when, now what has happened is that one of the eggs have, one of the sperm has managed to fertilize. Because one of the sperm has managed to fertilize, we cannot let any other sperm enter. We have to make sure that no other sperm is going to enter into your, no other sperm is going to enter into your egg cell. Otherwise, what will we get? We will get something known as polyspermy. Polyspermy is when multiple sperms will fuse with the egg cell. We don't want that. Because if multiple sperm will fuse with the egg cell, you will end up getting an abnormal chromosomal number. So to prevent polyspermy, to make sure that no more sperms can enter, you will get a cortical reaction, in which what is going to end up happening, that your zona pellucida, so the moment one of the sperm has managed to penetrate, the zona pellucida is going to harden. Okay, the zona pellucida is going to harden, and after that, the egg cell is going to start to produce cortical granules. They're going to start to react, produce cortical granules. What will happen? The cortical granules will then combine with the zona pellucida to make a tough membrane. And this tough membrane will make sure that no other sperm can enter. This is known as cortical reaction. And once the cortical reaction is done, the male nucleus and the female nucleus can fuse with each other to complete the process of fertilization. And now we have a zygote. Got it? Okay. Okay, Manu, we'll go ahead and stop it over here for today. We are basically left with our the topic of plants. Okay, so we will go ahead and we will do that in the other next class. Hold on one second.